Manjula, your voice not audible. I think, do you have any issue with mm, sound? Mic.
So now I am audible.
Yes, yes.
Okay, sir. Thank you. Can I explain the first question once again?
Yes, you can continue.
Okay. Thank you. So now I am audible. Fine, students. Let us go to the second question. So we have discussed it now only. The question is, the increase in the osmolarity of the outer to the inner medullary interstitium is maintained due to. In the statement 1, they are telling that a close proximity between the Henle's loop and the vasa recta. And in the statement 2, they are telling that counter current mechanism. In the statement 3, that selective secretion of bicarbonate and the hydrogen ion in the PCT. And in the fourth statement, they are telling that high blood pressure in the glomerular capillaries. Here you have to select which one is the correct statement. Okay. So in the option 1, they are telling that only 1 is correct. In the 2, they are telling that 3 and 4 is correct. And in the 3, 1, 3 and 1, 2 and the 3 is correct. And in the 4, 1 and 2 is correct. Which one is the correct? So you can select the answer, which one is the correct. Yeah. So let us answer to this question, guys. The first statement, they're telling that the close proximity between the Henle's loop and the vasa recta. Yes. So here you can see the osmolarity is maintained by the vasa recta, right? That is by the counter current mechanism. In that we can see the vasa recta, right? Vasa recta and the Henle's loop. That is correct. That is a maintaining the, here you can see this is the vasa recta, and here you can this is a this is a Henle's loop and this is a vasa recta. Here by the close proximity we can maintain the osmolarity. That is the correct statement. Then coming to the second statement, counter current mechanism. Yes, by the counter current mechanism we can maintain the osmolarity. Yes, that is also correct. Then coming to the third, they are telling that selective secretion of bicarbonate and the hydrogen ion in PCT. No, the osmolarity cannot be maintained by the HCO3 and the hydrogen ion. The osmolarity is mainly controlled by sodium, chloride, and the urea, and also the water is involved in it, right? So the mainly the ions involved in the osmolarity are sodium, chlorine, and the urea, right? And the water is involved in that. Whereas this bicarbonate and the hydrogen ion are involved in the maintaining the pH, right? They are involved in maintaining pH. Where maintaining the pH is different and maintaining the osmolarity is different. Okay, so that's why this third statement is wrong. Then coming to the fourth statement, the high blood pressure in the glomerular capillaries. No. So the high blood pressure in the glomerular capillaries, it is involved in the filtration of blood, right? So it is involved in the filtration of the blood in the Bowman's capsule. We can see that, right? So that's why it is not involved in maintaining the osmolarity, whereas it is involved where it is present in the cortex region, right? So that's why it is not involved in the maintaining the osmolarity in the medullary interstitial fluid, because it is present in the cortex region, and again it's not at all involved in the maintaining the osmolarity. Okay, it is involved in the filtration. Fine. So that's why in the option 1 they are telling only 1, no. Whereas in the 3 and 4, no. 1, 2 and 3, no. The correct answer is 1. Okay. That's why the correct answer is 4th. Fine. So let us go to the next, guys. In the urine formation today, we are going to learn about the counter current mechanism. Okay. So what is this counter current mechanism? Counter current mechanism. Guys, this is a very easy topic. Don't confuse here. Okay. So what do you mean by current first? Let us see, guys. Okay. The movement of the filtrate. So here you can see the movement of filtrate. Movement of filtrate. So where the movement of the filtrate is going to be happen, guys? Where the movement of the filtrate is going to be happen? It is going to be happen in the endless loop, right? The movement of the filtrate in endless loop, and the movement of blood, blood in vasa recta, movement of blood in the vasa recta, we will call it as the current. So how the fluid will move, right? So this is moving. So that we will call it as the current. The movement of the filtrate in the endless loop and the movement of the blood in the vasa recta. Okay. So then coming to the what is the current here? The current is what is the meaning by the counter? The counter is nothing but here you can see the movement in the movement of the fluid in the endless loop, that is just opposite to the movement of the fluid in the vasa recta, right? Here the movement of fluid in endless loop. Yeah, I will write like the HL. Okay, endless loop, and the blood in vasa recta. Blood in vasa recta is opposite to each other, right? Is opposite, right? Opposite to each other. So that's why we are calling as a counter. So here the filtrate is moving like this, whereas the blood is moving like this, right? So that's why we will call it as a counter current mechanism. Okay. So what will happen during this time? The ions will be exchanged from the ascending. Here you can see, guys, mainly the ions which is present in the, which is releasing from the ascending loop of the Henle, that is going to be present in the interstitial fluid, that is going to be absorbed by the descending limb of the vasa recta, and here you can see the fluid which is moving from the descending loop of Henle, that is going to be absorbed, that is going to be released in the interstitial fluid, where it is going to be absorbed in the ascending limb of vasa recta. So that is why we will call it as a counter current mechanism. Okay, fine. So let us, what is you mean by actually the counter current mechanism? We know that counter current mechanism, we will do it for the concentration of the urine. Okay, mainly it is involved in concentrating urine. So how it is going to be work? Let us see. Okay, when the blood was filtrated, it is filtrate, right? The urine or the blood was filtrated. On that time, the filtrate is having the concentration of 300 osmol. Okay, that is 300 osmolarity. So here you can see after it is coming to the proximal convoluted tubule. When it comes to the proximal convoluted tubule, it is a 300 osmol, and when it comes to the descending limb of the Henle, the osmolarity is 400. Okay, the osmolarity is 400, and you know that in the descending limb of Henle, here the mainly water reabsorption to be taken place. In the urine formation you have studied, right, guys? The reabsorption means the water is going to be reabsorbed into the interstitial fluid, right? It is going to be reabsorbed into the interstitial fluid, that is in the descending loop of the inlet. It is permeable, right? Permeable to the water movement. So now what will happen? When you remove the more water, it is going to be concentrated or diluting. Can you tell me, guys, whether it is going to be concentrated or diluting? When you remove the water from the salt, obviously it is going to be concentrated, right? For example, if you see salt water, so you mix the water with the salt and you are going to keep that into the outside. So in the sun, you are going to keep that. Okay. So what will happen, guys? The water is going to be evaporate and only the salt is going to be remain, right? The concentration of the salt is going to be more on that time, right? The same thing happening here. When you remove the water, the concentration of the salt is going to be more, right? That's why here you can see from the molarity is changing to the 600. And again, the water is going to be removed from the descending loop of inlet. Again, the concentration is going to be decreases. That is 900. Then again, the water is going to be decreases. Again, the concentration is reached to the 1200 osmol. Remember, guys, it is reaches to the 1200 osmol concentration. So now you can see only a more salt is there, right? That is the sodium chloride and the potassium is there now, right? So now what will happen when ascending in the ascending loop of Henle, the ascending loop of Henle is permeable to the electrolytes, right? It is not permeable to the water. Now what will happen in the ascending limb of Henle and the ascending loop of Henle, here the electrolyte is going to be removed. Electrolyte is going to be reabsorbed into the interstitial fluid. Here you can see, guys, these are the interstitial fluid. What do you mean by interstitial fluid? The fluid which is present between the two tissues, that we will call it the interstitial fluid. Okay. So here what will happen? The sodium chloride and the potassium is going to be removed. That is mainly sodium chloride. Okay. The mainly sodium chloride is going to be reabsorbed here. And here you can see when the sodium chloride is going to be reabsorbed, it is going to be removed from the loop of Henle, right? From the ascending loop of Henle. So here the concentration is going to be now decreases. Okay. So in the descending loop of Henle, the concentration was increases. In the ascending loop of Henle, the concentration is goes on decreases. Why? The electrolyte is going to be removed, right? So that is the reason. So see here from the 1200, it was reaches to the 700. And from the 700, again the sodium chloride and the potassium is removed, right? Again the concentration was decreased. When the salt was removed, obviously the concentration is going to be decreased, right? Now it is going to be decreased to 400. Then again it was removed. Again the salt is removed. It is reached to the 300. And in the distal convoluted tubule, you can see that, guys, it was reaches to the 300 osmol. And in the distal convoluted tubule, it will reach up to the 200 osmol. Up to 200 osmol, it can reach in the distal convoluted tubule, DCT. Okay. In the DCT, the maximum osmolarity, it will reach up to the 200 osmolarity. Okay. 200 osmolarity, it is going to be reached. And they may ask you the question in the exam like you. So in the way you can see the maximum, that is the maximum osmolarity was lesser compared to the interstitial fluid. You can see any body fluid that is having the osmolarity of 300 osmol. Okay. Lesser than the osmolarity of body fluid is having in the distal convoluted tubule. That is the 200 osmol. Remember that, guys. Okay. So that is the one part. So here in the distal convoluted tubule, that is for the 200 osmol. So here now let us talk about the counter current, how it is exchanging that with the vasa recta. Here you can see, guys, the vasa recta, you know that is formed from the efferent artery, right? From the efferent artery. So here we are seeing here the opposite of the ascending loop of Henle is the descending vasa recta. Descending vasa recta. So here you can see, guys, when the ascending loop of Henle releases the sodium and chloride in the interstitial fluid, interstitial fluid was nearer to the vasa recta. That is the descending vasa recta, right? Now the sodium chloride and the potassium will be taken up by the vasa recta. So in the vasa recta it was a 300 osmol. When it is taken the sodium chloride and the potassium, obviously the osmolarity was increased. Again, the sodium, potassium, chlorine was taken up by the vasa recta, and the concentration here, osmolarity is going to be increases in the vasa recta. Until here you can see the vasa recta, it was reaches to the 1200 osmol. Just opposite, guys. Just you can see opposite to opposite. That is a counter current, right? That's the reason. Here 300 is there, here 300 is there. So when it is going upward, where here you can see when it is going downward, it is increasing. But here also you can see that it is increasing, but the fluid is moving in the opposite direction, right? Fine. So here the sodium and chloride was again was taken by the vasa recta, and the concentration is increases and it is reaches to the 1200. And here you can see in the ascending limb of the vasa recta, in the ascending limb of the vasa recta, the concentration was decreasing. Concentration was decreasing. Osmolarity was decreasing. Why, guys? Here you can see the ascending limb is opposite to the descending limb of the right. So from the descending limb of the loop of inlet, it was decreased. It was releasing the water. The water is coming into the interstitial fluid, right? Now the water is taken up by the vasa recta and the osmolarity is going to be decreases. You can see that if I added the 1 spoon of the salt to the 1 glass of the water, or the 2 spoon of the salt to the 1 glass of the water, the concentration is more, right? How to decrease the concentration, guys? Either I will add the more water to this, right? So the same thing is happening. To decrease the concentration, I will add the more water. So that is the reason here the osmolarity is going to be decreases. See here, guys. The osmolarity is 900. The water coming from the descending loop of the inlet, it is absorbed by the vasa recta. Due to that, the osmolarity was decreasing in the ascending limb of the vasa recta. Okay. So here it is decreases 600. Then again the water is moving to the vasa recta. Again it was decreases to 400. Again the 300. Okay. So this is how both this vasa recta is maintaining the osmolarity in the interstitial fluid. Okay. Remember this. In the exam, they may ask you like this. In which limb of the vasa recta the sodium chloride is going to be absorbed? Can anyone answer me, guys? In which limb of the vasa recta the sodium chloride is going to be absorbed? Either in the descending limb or in the ascending limb? In the limb, remember. And in the which limb of the vasa recta the water is going to be reabsorbed? That is in the ascending limb of the vasa recta. Okay, remember that. And other than this, the urea is also placed. Urea is going to be removed from the collecting duct and it was released into the interstitial fluid. Okay, to maintain osmolarity of the interstitial fluid. And some of this urea will be come into this ascending limb of the inlet for maintaining the osmolarity. Okay, so remember that the urea is going to be, it will come to the interstitial fluid maintaining the osmolarity, and some of the urea will go to the ascending limb. Some of the urea will come to the ascending limb for maintaining the osmolarity. This is how the both this vasa recta is maintaining the osmolarity in the interstitial fluid. Okay, fine. So let us go to the, do you understood this, guys? What is the, do you understood this process? Counter current mechanism. Fine. So let us go to the urine formation, guys. That is let us go to the micturition process. So here what will happen in the micturition process? Micturition process is nothing but the release of the urine, right? So how this release of the urine is going to happen? Let us see. Okay. So when we are in the relaxed condition, so we don't have any urgency, or we can think that when we are in the relaxed condition, we can go and urinate. On the time, how this is going to be connected with the micturition? See here, on the time when we are in the relaxed condition, so the gradually the urinary bladder was filled, right? Urinary bladder was filled on that time. Here, the stretching of the urinary bladder. Here you can see the urinary bladder consisting of the smooth muscle. Smooth muscle. The smooth muscle consisting of the transitional epithelium. Transitional epithelium. This transitional epithelium consisting of the stretch receptor. This transitional epithelium consisting of stretch receptor. When the urinary bladder was filled, okay, when the urinary bladder was filled, whether in the relaxed condition, I am telling that when it is filled, it will send the impulses to the central nervous system. It will send the impulses to the central nervous system. In central nervous system, you know that autonomic nervous system, right? Autonomic nervous system. This autonomic nervous system consisting of the sympathetic nervous system, sympathetic nerve, and the parasympathetic nerve, right, guys? Parasympathetic nerve. One is a sympathetic and the another is a parasympathetic nerve. When in the relaxed condition, on the time this parasympathetic nerve is going to be activated. Okay. When the parasympathetic nerve was activated, it will carry the motor neuron, and the motor neuron is going to be reach the urinary bladder. So here it is leads to the contraction of the urinary bladder, thereby relaxing the sphincter. You can see the urethra is consisting of the two types of the sphincter. One is the internal sphincter. Internal sphincter. The internal sphincter is the involuntary, whereas the external sphincter is the voluntary. Okay. So here what will happen? So here it was making the urinary bladder to contract and relaxes the internal sphincter of the urethra. Okay, remember guys. So during the normal condition, what will happen? It is going to be activate the parasympathetic ganglion. The parasympathetic ganglion will carry the information to the urinary bladder, where it leads to the contraction of the urinary bladder muscle. Okay, the smooth muscle of the urinary bladder is going to be contracted now. Due to that, it is going to be by the contraction of the urinary bladder and by the relaxation of the internal sphincter, where the urine is going to be released. Okay, that is during the condition. What will happen if the class is going on? We are not able to ask the teachers, can we go for the urination? On the time, what will happen? So here, the micturition process is going to be inhibit on that time, right? So on that time, remember guys, when during the inhibition of the micturition process, stopping of the micturition process, sympathetic nervous system, sympathetic ganglion is going to be work on that time. Okay, the sympathetic ganglion is going to be carry the information. Here it is going to be dilate the urinary bladder. It will dilate the urinary bladder. Thereby, it will lead to the contraction of the sphincter. When the contraction of the sphincter is happened, on the time urination is not going to be happen, right? It will lead to the contraction of the urinary contraction of the urethral sphincter, that is the internal sphincter. Due to that, it will inhibit the micturition process. Okay, so that will it will contract the external urethral sphincter. It will contract the external urethral sphincter. Due to that, the micturition process is going to be inhibited. Okay, remember this guys. When in the relaxed condition, parasympathetic is going to be work. It leads to the contraction of the urinary bladder and relaxation of the internal sphincter. Then coming to the in urgency condition, it is going to be inhibit the micturition process. On that time, it will dilate the urinary bladder and it will lead to the contraction of the external urethral sphincter. Okay, fine. So you can see that the neuronal mechanism that is causing the micturition is called as the micturition reflex. Here it is going to be controlled by the neuronal control, right? So that we will call it as a micturition reflex. Fine. So let us talk about the urine. You know that we know that the filter rate is going to be about the 125 ml per minute, 125 ml per, right? And it is about the 180 liter per day, right? Then how can we release only 1.5 liter, guys? 1.5 liter of the urine per day. Per day, we are going to release only 1.5 to 1.8 liter of the urine per day. You know that 99% of the filtrate is going to be reabsorbed, right? Only 1% we are going to be released. That is nothing but the 1 to the 1.5 or 1.5 to the 1.8 liter of the urine is going to be released. That urine consisting of the excretory waste, that is the 25 to 30 gram of the urea, right? The urea is the excretory waste in the humans, right? So it is going to be released by the urine. And urine is, you know that urine is a light yellow in color. Whenever the urine is more concentrated, on that time it will be dark yellow. And during the disease condition also it will be dark yellow. And the pH of the urine is the acidic, that is slightly 6 pH. And it has the characteristic that odor is the strong ammonia-like odor. And the yellow color of the urine is due to the presence of the pigment. That pigment is urobilin. Remember this, guys. Okay, urobilin pigment. Due to that, the urine is yellow in color. Okay, fine. So let us talk about the whenever the any disease condition is there, on that time the doctor will tell us for the blood test or the urine test, right? What is the reason actually? So by the blood test and the urine test, you will come to know about the what are the disease you are having, right? So here you can see by the blood test, you can see the glycosuria, whether the blood is, where the glucose is present in the urine or not. When you take the example of the diabetes, for the diabetes test, they will check for the blood also and in the urine also for the glucose, right? In the urine, the glucose concentration is more, means it will tell about the kidney is not functioning properly, right? Yes. Here you can see, to know, for the testing the glucose in the urine, it is having the test, that is the Bendick test for the glucose. Okay, not in the urine only, the glucose test is the Bendick test. Here you can see the glucose is more. The glucose is more in the, sorry, glucose is not at all there in the sample, means it will lead to the blue color. If the glucose is only a little, a trace amount of the glucose is there, means it will lead to the green color. And it is a moderate amount of the glucose is there, means it will give rise to the orange color. And the more glucose is there, more glucose there in the sample, means it will give rise to the a brick red color. A brick red color will tell that the more glucose is present in the sample. Okay, and the more glucose is present in the urine, it will tell about the diabetes. Okay, diabetes mellitus. Remember that, guys. Fine. I think you understood this. Do you understood this, guys? Tell me whether you understood. Fine. So let us go to the next. The role of the lungs, liver, skin in the elimination. So that is the excretion. So what are the roles, guys? Only kidney is involved in the excretion. Why this lungs, liver and skin is not involved? It is involved, right? So let us learn about this, guys. The lungs. You already know that the lungs is involved in the respiration process, right? So it is involved in the breathing. So during the breathing, what will happen? So we'll take the oxygen and we will remove the carbon dioxide, right? Same. So the lungs is involved in the removal of the carbon dioxide, okay, and the water. Then coming to the liver, you know that, guys, liver will secrete the bile juice, right? The bile juice is helping in the emulsification of the fats, right? So here you can see the bile juice is having the containing the pigments that are the bilirubin and the biliverdin, and it is having the cholesterol. So these will degrade the steroid vitamins and the drug, right? So this will, by the bile juice, this will carry to the duodenum region, and along with all the food is digesting, right? Along in the food is digesting in the duodenum, then it will come to the jejunum, then to the hilum, and then then it will go to the large intestine. To the large intestine, it will store in the rectum and it will remove through the anus, right? While some of the bile juice will be removed. So by this, although some of the waste material will be excreted through the bile juice, through the alimentary canal. Okay. Then coming to the next one, that is the skin. We all will sweat, right? Whenever you go to the high temperature, we'll sweat, right? So in that time, the sweat is consisting of the water, mainly water and the sodium chloride, and a small amount of the urea will be there, and the lactic acid also. Remember, guys. And the main function of the sweating, guys, why we will sweat during temperature? That is homeostasis, right? To cooling the body, right? So that is also the main reason. The sweating. Due to the sweating, we will cool the body surface, right? Then coming to the, we will release the sebaceous gland. The sebaceous gland will eliminate the oil, right? You can see that oily things will be come out from the skin, right? So that will be released by the sebaceous gland. Okay. So whenever it is released, it will release as the hydrocarbon, waxes through the sebum. Okay. The sebum, we know that the sebum will protect the protective oily covering for the skin. Then coming to the saliva, saliva eliminates the small amount of the nitrogenous waste, that is a urea. So you know that the lungs is involved in the removal of the carbon dioxide, liver is involved in the removal of the waste through the bile juice, through the alimentary canal, that is alimentary duct. And coming to the skin, skin is involved in the removal of sweat. In the sweat, the sodium chloride is going to be removed and a small amount of the urea. And the sebaceous gland, the oil is going to be removed through the sebaceous gland, and the saliva is going to be removed the nitrogenous waste. Remember this, guys. Then coming to the regulation of the kidney function. How the regulation is going to be happen? See here. Yeah. Whenever we are thirsty, we will drink the water, right? Is that we are doing simply? No, it is the one of the regulation process. Right? Okay. So we will see, guys, what are the things mainly involved in the regulation of the kidney. First one is the feedback mechanism, guys. Here it is involving the hypothalamus. The mainly here, remember, guys, the mainly it is involved in the hypothalamus. Hypothalamus is involved in the regulation. Hypothalamus, you know that whenever you are thirst, the thirst is going to be regulated by hypothalamus, right? The hypothalamus is controlling the thirst, right? And the other things like the juxtaglomerular apparatus and the aldosterone, and the ADH and the ANF. Okay, atrial natriuretic factor. This all the things is going to be regulate the kidney functions. Let us see this. Okay, fine. First, let us see, guys. If the blood volume was increased, there is a increased blood volume. 1 minute. Yeah. Blood volume was, let us go to the 1 minute. Yeah. Blood volume is decreased in the blood volume. The decreased in the blood volume obviously will lead to the decreased in the blood pressure. Blood pressure is going to be decreased, right? And it will lead to the decreased glomerular filtration rate, decreased in the glomerular filtration rate. And due to this all the thing, there is increased osmolarity, right? That's what will happen now, right? The dehydration, water is not there in the body. So now what will happen when the body fluid was decreased? So here, so there is it stimulates the water reabsorption. Here, the hypothalamus, guys, from the hypothalamus, the anti-diuretic hormone. Here you can see, 1 minute. Hypothalamus, the hypothalamus is having the osmoreceptor that is going to be activate the posterior pituitary. It is going to be stimulate the posterior pituitary. From the posterior pituitary is the ADH hormone, anti-diuretic hormone. What do you mean by diuresis, guys? Release of the more water, we will call it as the diuresis. Frequent urination, that we will call it as the uresis, right? Diuresis. Here what will happen? The release of the anti-diuretic hormone. The release of the anti-diuretic hormone, it is going to be reabsorb the or retain the water absorption. It is going to be absorb the water. So water from the DCT, that is the distal convoluted tubule and the collecting duct. You know the distal convoluted tubule and the collecting duct having the receptor for the anti-diuretic hormone, right? Because of this, the water is going to be retained. Water is going to be reabsorbed in the distal convoluted tubule and the collecting duct. So when the water is going to be reabsorbed, on the time what will happen, guys? Water is absorbed. Obviously, the volume is going to be, the blood volume is going to be increases, and the blood pressure is going to be increased. 1 minute. Yeah, it is leads to the increases in the blood volume, and it is going to be increases in the blood pressure, right? So thereby it is going to be decrease the osmolarity, thereby it is decreasing the osmolarity. Then it will be a normal condition, right? And you know that, guys, when this ADH hormone is not there, so deficiency of this ADH hormone will lead to the diabetes insipidus. What will happen in the diabetes insipidus? A frequent urination is going to be happen. Why? The ADH hormone is not there, right? To where the ADH hormone is there means it will reabsorb the water from the DCT and the collecting tubule, right? Collecting duct. But here the deficiency of ADH will lead to the diabetic insipidus. Okay, the frequent urination is going to be happen. Okay, but the urine is not having the glucose. Okay, diabetes means It is not in the diabetic insipidus, the urine is not having the glucose. Okay, remember that. Fine. And remember, guys, it will lead to the vasoconstriction. This ADH is the vasoconstrictor. Vasoconstrictor. Okay, fine. So let us go to the next thing, guys. Fine. So here we'll go to the RAS mechanism. What is the RAS mechanism? So here you can see RAS, we will call it as RAS. What is the full form of the RAS, guys? RAS is renin, angiotensin, aldosterone, renin, angiotensin, aldosterone mechanism. Okay, what will happen when it is going to be activated, guys? When the blood pressure is going to be decreases, when the blood pressure decreases, when the blood volume decreases, when the blood volume decreases, okay, and when glomerular filtration rate decreases, on the time this RAS mechanism is going to be activated. And you know this, guys, the RAS mechanism is a powerful vasoconstrictor. It is a powerful vasoconstrictor. How it is going to be activated? Let us see, guys, here. Okay, so here, see here, the liver is going to be produced one of the enzyme that we will call it as a angiotensinogen. Remember, guys, whenever the gen is there, it is the inactive. Okay, and from the kidney, the renin is going to be produced from the juxtaglomerular cell. The renin is going to produce juxtaglomerular cell that is going to be activate angiotensin that is released from the liver to the angiotensin 1. And in the lungs, this angiotensin 1 is going to be converted from the lungs. ACE is going to become, the ACE is going to be activate this angiotensin 1 to the angiotensin 2. Okay, so here the angiotensin 2 is going to be act on the adrenal gland. It is going to be act on the adrenal gland and it will release the aldosterone. Aldosterone is going to be released, guys. What is the main function of the aldosterone? The aldosterone is going to be reabsorb the sodium and water from the distal convoluted tubule. So that is from the distal convoluted tubule. Okay, that is the one process. When the water and the sodium chloride is reabsorbed, obviously the volume of the blood is going to be increases the blood volume, right, and increases the blood pressure and the glomerular filtration rate, right. And here you can see, other than this, the angiotensin is going to be, this is also going to be act on this aldosterone or this activity, this RAS mechanism is also going to be act on the proximal convoluted tubule, and it will lead to the absorption of sodium chloride and water in the proximal convoluted tubule, where it is releasing the ADH. Okay, 1 minute. Yeah. Due to this RAS mechanism, the ADH is also going to be activated. This aldosterone is going to be activated, the ADH. Okay, fine. Remember this, guys. Okay, and in the proximal convoluted tubule also, the sodium chloride and water reabsorption is going to be taken place. This is about the RAS mechanism, and it will lead to the increase in the blood volume, increase in the blood pressure, and the glomerular filtration rate. And sometimes what will happen, this RAS mechanism was increased more. Due to this, the blood pressure, blood volume is increased. Due to that, it is going to create a pressure on the heart. Okay, so creating the pressure on the heart wall. Due to that, the heart wall is going to be produced the one of the factor, that is the atrial natriuretic factor. That is atrial natriuretic factor. Okay, this is released by the walls of the heart. Where it is going to be released, guys? Walls of the heart. Walls of the heart, it is going to be released. It is going to be inhibit the RAS. It is going to be inhibit RAS mechanism. It is going to inhibit RAS mechanism. When it inhibits the RAS mechanism, obviously it is going to be decrease blood pressure. It is going to be decrease blood volume, and it is going to be decrease. It is some normal condition of the glomerular filtration rate. Okay, so this is how it is going to be for the work. But remember, guys, The ANF is a vasodilator. Vasodilator. It is dilating the blood vessel. Coming to the RAS mechanism, aldosterone and ADH. These three are vasoconstrictor. Only one is a vasodilator here, that is the ANF. Okay, remember this. Fine. Coming to the disorders of the excretory system, guys. So here the disorders are, first one is the uremia. So what do you mean by uremia? When the urea concentration, when the kidney is unable to filter the urea or malfunction of the kidney will lead to the accumulation of the urea in the blood. Okay, accumulation of the urea in the blood, that we will call it as the uremia. Okay, that is due to the mainly kidney failure. This kidney failure can be used, the person who is having the kidney failure will be used the dialysis process. Okay, fine. So let us go to the renal calculi. So here you can see, guys, this is a renal pelvis, right? This is a funnel-like structure, that is the renal pelvis in the kidney. So here what will happen, the oxalate, that is the oxalate is going to be accumulate more and it is going to be form a stone. Okay, that stone we'll call it as a kidney stone. That mainly the kidney stone is going to be formed from the oxalate. It is going to be accumulated in the renal pelvis. So that's why it is called as a renal calculi. Okay, remember that. Here you can see the different sizes of the kidney stones that is recovered, removed. Fine. So let us go to the glomerulonephritis. That is, glomerulonephritis is nothing but the inflammation of the glomeruli. So here you can see this is a healthy kidney. Glomerulonephritis. Here you can see the inflammation in the glomerulus. Okay. Fine. Let us go to the last one, that is the hemodialysis. Hemodialysis, we also call it as, guys, artificial kidney. Artificial kidney. Okay. So what they will do in the artificial kidney is, guys, so here they are going to be going to cut the radial artery. That's the mainly artery. Here you can see the artery, though they are going to cut, that we will call it as the radial artery. Radial artery they are going to cut, and here is the vein is there. Okay, so they will cut this radial artery. From this the blood is going to be moved. The blood is rich in the urea, right? And here they will move that urea, that they will move that this blood into the blood is coming from this, that is they are using here is the cellophane tube. Here you can see the cellophane tube. From the cellophane tube, it is going to be carry the blood. That blood carrying from the cellophane tube, that is carrying the blood, that will be passed into this tube. Here you can see that is the dialysis chamber. Dialysis fluid. Here you can see this consisting of the dialysis fluid. Here you can see this is the dialysis fluid. Before passing to this dialysis fluid, here they are adding the blood. For the blood, they are going to be add the heparin. So that is the heparin. 1 minute. So here they are adding the heparin. Then they are passing this blood through the cellophane tube to this fluid, where this fluid is having the high concentration of all the things, but it is having the isotonic. This is an isotonic medium. Isotonic medium. It is having the isotonic medium for all the electrolytes, but except the urea. Okay, but except the urea. Except urea. Okay, so now what will happen? The urea which is present in the blood, obviously it will move through this to the fluid. Okay, it will move from the higher concentration to the lower concentration, right? The fluid is having the lower concentration. Now the blood will move from the fallopian tube to the cellophane tube to the fluid, and now this urea is going to be collected here. Okay, and now this blood which is going out from the from this fluid, it is having the no urea. Now for this now they are adding the antiheparin. Now they are adding the antiheparin. Now this blood will be passed through this vein. Okay, now this blood will be passed through the vein. It is going to be go to the all the parts of the body. Okay, remember, guys. So this is how they are doing the dialysis. Fine, let us go to the kidney transplantation. You know that if the person is having the kidney failure, for them the kidney transplantation will be done. The kidney transplantation will be not done from the, not everyone will use the kidney. Okay, kidney should be matched. For example, before they will do the blood matching. Okay. First they will test the blood. Blood should be matched, and also the tissue should be matched. Tissue should be matched. And from the close relative, the kidney will be taken from the very close relatives. Okay. And for them, the blood will be, tissue will be matched. So that's the reason the kidney transplantation will be done. Okay. And here, if it is why this kidney transplantation will be rejected by the other people, if the other people given the kidney, it is not matching the blood and the tissue, means it will be rejected by our immune system. Immune system is going to be rejected. That is mainly cell mediated immune system. Cell mediated immune system. Cell mediated immune system is going to be reject the kidney. Okay. And here there are so many drugs is going to be used for the for the suppressing the immune system. Obviously, if the immune system is suppressed, means obviously the rejecting percentage is going to be decreases, right? So that is the reason the immune system is going to be suppressed by using the drug cyclosporine. Cyclosporine. Some of the drug like cyclosporine is used for suppressing the immune system. Suppressing immune system in the kidney transplantation. Okay, fine guys. So that's it for today. If you have any question, guys, you can ask me. Janvi Varda, if you're having any question, you can ask me. Fine, guys. Let us wind the class. Thank you so much. If you have any questions, means you can ask me. Otherwise, we'll wind up the class. Thank you so much, guys. Let us meet in the next class.